I want to welcome everybody to the International Alliance for Phytobiomes Research webinar series. We're joined today by Dr. Karsten Zengler, who's going to be talking to us about Rhizosphere microbiomes. Before we get into that seminar, I just want to introduce a few things about this webinar. And the first is that it's, hold on a second, being sponsored by the International Alliance for Phytobiomes Research, and this is a nonprofit consortium of industry, academic, and governmental scientists. And this is an alliance that anyone can become a member of simply by being involved in projects or by becoming a sponsor. You can see that we have sponsors that range from academics to many different companies that are interested in various aspects of plant system research and phytobiomes. Just in case people aren't as familiar with this as they might be, I wanted to introduce the concept of phytobiomes. People often think of it simply as plant microbiomes, which are in fact a very important component of phytobiomes and one of the more complex components, but a phytobiome itself goes way beyond the microbiome in that everything that the microbes are doing to the plant or with the plant or in the system is influenced by all the other factors, including surrounding plants, all the geophysical factors of the system, like the climate or the weather, the other arthropods or animals or plants. And it's when all of these interact together, and in an agricultural setting, you're layered on that with the management practices that are undergoing, that you have interactions that are affecting the outcome and the functioning of these plant-based ecosystems. And what we realized when we were really forming the Phytobiomes Alliance is that people have been quite siloed in looking at individual aspects of these phytobiomes, which has been critical, but there is a point at which we have to be integrating all of this information. And even as individual researchers, when we work on one component of the phytobiome, we always need to keep in mind how it's interacting with other components of the system. So we think broadly about phytobiomes. They are definitely relevant to all aspects of agriculture, field crops and vegetables crops, but they're also relevant to forage crops, so pastures, to forest health. Those are a major type of phytobiomes that are relevant to the planet, but also all the new types of indoor agriculture, like vertical farming, that are coming on. All of these involve making decisions about what plants can grow in that system and what are all the components of the abiotic and biotic environment that are influencing how those plants grow, how that whole ecosystem is sustaining it and being maintained over time. So the vision of the Phytobiomes Alliance as a group is that by 2050, all farmers will have the ability to use predictive and prescriptive analytics that are based on a real comprehensive understanding of the geophysical and biological conditions that are present in a particular phytobiome to help determine the best combination of crops, management practices, and inputs for a specific field in a given year. The Alliance sponsors a number of different events, and I just wanted to highlight a couple that are upcoming. These are both at the International Congress of Plant Pathology in Lyon, France. The two workshops that the Phytobiome Alliance is sponsoring are the Harnessing Culture Collections for Improved Plant Health. This is for both formal collections and informal collections in individual labs or companies. And also an afternoon session that looks broadly at different phytobiomes research for plant health. You can at any time register for these workshops or register to the mailing list to receive news about any of the events organized by the Phytobiomes Alliance. So just a few mechanics. This webinar is in fact recorded and will be posted on the Phytobiomes Alliance YouTube channel later today. You can subscribe to the channel if you never want to miss a new upload. The presentation for today will be followed by a question and answer session. At any point before, during, or after, you can submit your questions into the Q&A panel. You can see the question bubble that you can click on. Organizers will be posting things in the chat, things like websites or maybe papers that are referenced by Dr. Zengler, so you can monitor that chat panel. And you can also obtain the handouts, and that meaning you can download the PDFs of the presentation at any point as well. So with that, I am really happy and excited to see a presentation by Dr. Zengler. He is coming to us from the University of California, San Diego as a professor in pediatrics and bioengineering. Dr. Zengler has his PhD from the Max Planck Institute for Marine Microbiology in Bremen, Germany. He has worked in originally in the high throughput cultivation of very difficult to cultivate organisms, but he has 30 years of experience in combining microbial physiology, molecular biology, computational and systems biology to really understand microbial communities and the roles of microbiomes in health and in disease. So with that, I turn the microphone over to Dr. Zengler, and I thank you for your participation today. Just a reminder, I will come back at the end to help moderate the session, the questions.
Okay. I hope you switched over the presentation. Can you see the presentation? Show my screen. Yep. Okay. So thanks, Gwen, for the nice introduction. I think I can skip a bunch of my intro slides based on this, because we study the microbiome in the context of plants. What I present today will be mainly about the microbiome itself, so the bacteria, but our effort is currently also on the plant side. So if you give me another half a year or year, this presentation will include a lot of plant data as well. Okay, so to start off, just a few disclosures. So I have a problem to advance this. Yeah. So I'm a co-founder of a couple of companies and SAB member of different companies related to human mainly efforts, but also in animals, and we do the same now for plants as well. Okay, so as you are probably all aware, we have made a lot of progress in microbiome science in general over the years, but this process or this progress is not really even distributed among many different aspects. For example, and this is my personal view of the field, and as Gwen said, I've been in the field for around 30 years, so I have a good perspective, I guess, when it comes to what we have achieved as a community in microbiome research. So based on the improvement of sequencing data, we know pretty much now who is out there, from the organism level down to the genome level, because we can sequence very deep and very cheap right now. So who is there? There's a big improvement over the years. What are they doing? This becomes more questionable, because many of our biochemical data, we can do this in bulk, but we often cannot relate this to individual species. How they are doing it, let's get even more sketchy, because we rely on a lot of biochemical data, and biochemistry hasn't been well funded over the last 30 years. So this is a lot of pure culture work, and we infer most of our data from a couple of model organisms. And then why they are doing it, I think this is out to debate, because most of the time we do not know why an organism is carrying out a certain function and not another function at a certain moment in time. Okay, so this is actually related to the microbiome, in this case of the rhizosphere. So I put a couple of pictures of different trees up there to highlight different environments. And we have done this extensively over the years as a community, but we still don't know from our, for example, sequencing data, we cannot infer who else lives in this environment. We have data, but we cannot, for example, transplant a plant from one environment to another because the data is not suitable to generate any information about this. we can definitely not build or create environments from scratch. People have tried this, but this has been so far a failure, and we cannot predict anything what happens in the environment to a detailed way. Okay, this, what I said, is not only true for terrestrial and the phytobiome, but it's also true for every kind of microbiome we have studied. So this is true for aquatic as well as terrestrial microbiomes, and of course all the host-associated microbiomes, which includes the human as well as plants. So what we have failed right now, these big questions in microbiome research, is how do communities respond to perturbations? And we can also not right now predict outcomes of perturbations very well. So these two open questions we try to address, and I hope I will give you some glimpse into this, how we have done this and what progress we have made over the last, I would say, 3 to 4 years in this area. Okay, so if we address both of these questions, how communities respond to perturbation, and if we can predict anything of this, we can actually control the biomes. We can change them towards making the microbiomes more suitable for plants, for example, to increase yields or increase drought resistance, and we can rationally design these microbiomes to our needs. So the presentation I will give will mainly focus on the process in this rhizosphere, and we have done a lot of work on this. But first, I will start giving an overview of how we study microbial communities. So this is both an overview what people have done in the past, and also you will see that there are certain shortcomings in our studies of microbial communities. Okay, so this is a collection of most of the processes out there, how we study any kind of microbial community. So on the left, you have the communities, and people study either the DNA, the mRNA, proteins, or metabolites. And on the right side, you see there's a couple of techniques out there. So you do either amplicon sequencing, so RNA, 16S RNA sequencing, or 18S for fungi, or ITS for fungi, and you look who is there. We have done genomics as a community, like a lot of shotgun metagenomics, so then you get an idea what is possible and study the genes. You can do transcriptomics, so look at the transcription, and so you try to understand what happened this moment in time. Same is true, people have done proteomics or metabolomics, where you measure all kind of metabolites. If you look over the last roughly decade, all the articles we've published in microbiome research, you see that over 90% focused on either 16S DNA or metagenomics, so by identifying a footprint of an organism. Very few work has been done on metatranscriptomics, proteomics, or metabolomics. So, so far, this gave us a good idea who is there, but most of the microbiome research is still very correlation-based and descriptive. So this funny little cartoon here highlights what happens, how you study different things, and you know if it's hot weather right now in July, most of the places you guys are probably chiming in from, it is pretty warm and hot, and you know you go outside, you get a sunburn, so there's a clear causation, and people go out and buy a lot of ice cream. However, just because you buy ice cream doesn't mean you get a sunburn. So the correlation, the causation between certain effects, it's not very clear, and often microbiome research tries to link, in this case, sunburn and ice cream together, which the outcome can be questionable. Okay, so we set out to make microbiome research not only correlation-based, but we want today make microbiome research really causative, so we want to establish causation, and we want to make it predictive, so we can make predictions of the microbiome, how it behaves. So coming back to the slide, if you look at the slide, what really defines the microbial phenotype, so how do you go from the DNA to a phenotype, the most, you go from a gene to mRNA to a protein, so transcription and translation. However, there is not a method out there which is commonly used to study translation, which is kind of surprising because translation is by far the most expensive process in the cell. So we have ignored translation for a long period of time, although the cell has to spend the most energy and money, or ATP, NADPH, on this process. It's also extremely tightly regulated. So how to go from the DNA, from a gene, to the mRNA, to the protein is very tightly regulated inside the cell. And this regulation is done by translational efficiency. And I will explain this in a couple of slides, what is meant by this. But how do you study translation? So there has been a method out there, which is translatomics, so a riboseq. And we have used this in the past to study the translation efficiency of single organism. So in a nutshell, what is riboseq? Basically, what you do is you have a sample, you stop the translation, so the ribosomes in its tracks, you digest everything around the ribosomes, so you end up with mRNA, which is sandwiched here within a ribosome. You remove the ribosome, and then you sequence these tiny little fragments here inside the ribosome. So if you know where the ribosome is on an mRNA and what is translated at this moment in time. Okay, it's pretty straightforward. Has some ins and plus and minus of this method, but this has been done for single cells. Coming back now to translation efficiency. So if you do these translatomics, you can study how efficient the organism translates and transcribes the gene from going from a gene to a protein, right? So in this case, you have these orange mRNA or DNA. You transcribe it, you translate it, you make a protein. So a little mRNA gives you a little protein. A lot of mRNA gives you a lot of protein. However, since I said earlier, there's a lot of regulation involved, the cell very, very tightly regulates this. And there are many cases where you make a lot of mRNA, but the translation efficiency is very low because you make a lot of mRNA, gives you very little protein, or you have opposite cases where you have a little number of mRNA molecules, but they get translated extremely efficiently and high at this time, and you have a very high translation efficiency. So what does this relate to? So basically, the cell controls phenotype through translation efficiency. So it allocates this resource. And this is a good analogy to how we normally allocate our resources. So most people here on the call have a finite number of money, and you have to spend the money for groceries, housing, transportation. You maybe buy jewelry, go out for fancy dinner, go on vacation, and party. So now inflation is rampant, around 5% in the US right now, much higher also in other countries around the world. And you can see that now, for example, grocery prices and gasoline prices go up a lot. So, however, if you're really inclined to go on vacation, you will rebudget and reallocate all your money in order to still afford vacation you want to go. So the resource allocation defines a person's preference. The microbes are not much different in this sense, that they have a finite amount of energy available, and they have to do all these different things, from synthesizing sugar, dividing, making DNA, making cell membranes. And this cell, for example, prioritizes its resources by swimming around and eating fatty acids. Okay, so resource allocation in a bacteria defines the cell's preferences. So after we have done this, we thought, if we know what the cell's preference is, we can classify all these microbes and figure out who is doing or wants to do a very similar function. And so this is called a guild. And I will go over this in a second in much more detail. Okay, so the translation efficiency is determined by looking for all the translation compared to all the amount of mRNA you have available. And one of the trick we have done here is we moved this method from a single organism to a community. So we do meta-RNA-seq and meta-riboseq. And then the hope is that we can really control, change, and design community by understanding what these organisms want to do. So first we started off with validating our method. And for this, we use the synthetic community instead of going into soil right away. So we use the synthetic community, which we designed of 16 members from the switchgrass Rhizosphere. So these are organisms which are commonly found not only on switchgrass, but on a bunch of different grasses. And we generated this community. It's very stable and robust. And there's a manuscript down here you can look up how we describe this. And we deposited all these strains into DSMZ so everyone can use them and generate information, which then we can build on as a community. So this is really like a community effort where we try to have defined model system for a Rhizosphere community. Okay, so we use these 16 strains. We grow them all up. We put them in the lab in some medium. We grow them for 7 days in a bunch of replicates, and then we performed metagenomics, so looking for the DNA, metatranscriptomic, looking for the mRNA, and metatranslatatomics, looking for these ribosome footprints. Okay, so this is the first data we generated, and you can see here the metagenomic data, metatranscriptomic, metaribosic data. Within the replicates, they look pretty similar. These are the different organisms. However, if you put them in a PCA plot, you see that they all cluster nicely together. So the metatranscriptomic, metagenomic, metaribosic data cluster nicely together. However, they are very distinct from each other. This is good because this means that these organisms actually do different things and how they read out the DNA to mRNA to translation and then proteins. Okay, so we developed then a method which determines the guilds and the microbial niche determination, which we coined MIND. So how did we do this? So we used all the TE, what I just explained to you before, how this is done. And we used this TE and basically clustered these organisms based on all their pathways and see which organism do the same kind or want to do the same kind of thing, right? So what is the resource allocation of these microbes and how do they differ from each other? And you can see if you put them in these dendrogram here that you get 6 different clusters, and within each cluster every organism, that's what the assumption, does something else. Okay, these guilds based on the translation efficiency are very different than what you get from phylogeny. This makes sense because not everyone who has the same name does the same kind of function, and you can see that these organisms, the prioritization, so what they really want to do in life, is different than their heritage based on the 16S level. Okay, there's also different not only 16S level here on the right, but also on the genome itself. So the presence and absence of all the genes in the genome looks very different than the clustering based on our guilds based on the translation efficiency, as it not the same as a riboseq data and not the same as a RNA-seq, so the transcriptomic data as well. So this is really some unique information, and this clustering differs. So does it make any sense? I'll explain this to you hopefully in a couple of slides later. But we also, one point I want to make is that these prioritization of pathways, or what the cell really likes to do, is very different if it grows when it grows by itself in axenic culture or if it grows in a community. And this is data from here 1, 2, 3, 5 different isolates where we grow them together in a community, so like in the member of 16, or we grow them individually. And you can see from this histogram here that between these organism grown axenically as an isolate or in a synthetic community, it really prioritizes different things. So the organism wants to do something different when in a community than in an isolate. So this means that the organism prioritizes different pathways when in a community. Okay, so the question we had then now, can our guilds, which we generated, really predict the intervention outcomes? So this was the goal of the study. So the first thing is since we had a synthetic community, we said, how about we just remove this organism and see what happens? So we did this. Here's your guilds. We removed this organism Mutalligine-Bacter. This is a control, what happens. So now you remove Mutalligine-Bacter, and that how the abundance then looks later. And you can see from this figure, I explained it in a second, that when Mutalligine-Bacter is removed, so this triangle is down, one organism, which is Cyanophaga, and this organism, Burkholderia, they both go up in abundance. So what is printed, shown here, and I will show a bunch of these figures, is the abundance of these organisms in the syncom against the abundance of the organism in the syncom with the modification, in this case, the removal of these Mutalligine-Bacter. And the dark gray spot here is a 99.9 confidence interval. So this is your everything which is outside this bar, you are 99.9% confident that this is the real data point. So this is all done in triplicates. So that's where the statistics come in. Okay. However, one of the things, it's often difficult to have these clusters two-dimensional, because we know that this is actually not the case. They're actually arranged in a 3D version. And so we try to identify competition scores. So this means we calculated the distance from each cluster, from each organism to another organism, based on their distance in the 3D space. It's like a z-score, and then we came up with a matrix where we have these organisms here, and if they are red or yellow, they have a high competition. If they're blue, they have a low competition score. Okay, very easy. Red means they don't like each other. Blue means they're very happy together. Okay, so we did this then for a bunch of organisms, where we always removed an organism and could see if we can predict what will happen. So out of these 16 members, we remove, for example, Arthrobacter, and you can see here Arthrobacter is gone, and now we increase these two different organisms, the purple one, which is Brevibacillus, and the orange one, which is Cyanophaga. And both of them we are able to predict a priori with a competition score that these organisms will actually increase. Okay, we did this for another organism, Lysobacter. We removed it, two organisms went up, and again the competition score predicted that these two organisms likely will increase in abundance. And we did this for a whole bunch of organisms, and every single time we can show that the organism which will increase based on the removal of an organism is indeed predicted by our competition score. So this means we are 100% sensitive in this analysis, but 74% specific. Okay, good. So we were very excited about this and said, this means likely that these bacteria who are in the same guild are actually competitors. Every time we take one away, another one in this guild takes over. So, and this was the case because we also looked at in more detail what kind of competition they are actually performing. And one of these clusters, these guilds, Cyanophaga and Mutalligine-Bacter, when we looked at all the prioritization, what they did, they had a lot of genes which were looking that they were not very happy with each other. So they had antimicrobial peptide resistance, they produced beta-lactam resistance, they produced other toxins and drug resistance. So they looked like these organisms were actually really putting on the gloves and having a fight in this community. So in order to prove that this has happened, we did a spot on lawn assay. So you take these strains, you put them on an agar plate, and then you spot here on this lawn, you spot another organism, and you can see if you get a clearing zone, which means, aha, these organisms don't like each other. Someone is producing a small molecule, an antimicrobial, in order to fight these other organisms off. So a very classic test. So we did this, and what I show here is a lawn of all these 16 different bacteria, and then spotted on here is the Cyanophaga in the middle as E, and you can see if you look at this spot G here, you can see a clearing zone. If you zoom in, you can see the clearing zone is pretty defined. So this organism is producing something against these other organisms. And if you look who this other organism is, Cyanophaga really produces something against Mucinigenibacter. Strikingly, it only produces a molecule which works against these Mucinigenibacter, not against any other of the members. And likely because there is no need to fight any of the other members because they have very different priorities. They carry out very different functions in this community. So there's no need to get angry with any of the other organisms outside this guild. So you only produce antimicrobials which fight specifically your direct competitor. Okay. So then we said, so how about now we can play around with this and we add members to the community. So this is like a probiotic intervention. And so what we did first, this is here again our clustering. I showed you before we moved Burkholderia and we could see, so the triangle goes down. We increase specifically Rhizobium, an organism in the same guild as Burkholderia. And then we said, okay, what happens now if we add Burkholderia? So we add Burkholderia and we can see that its competitor Rhizobium is reduced in abundance. Okay. Then we said, how about we add Rhizobium? We add Rhizobium in higher amounts to this community. We can see specifically Burkholderia goes down. We also remove both organisms and we see it has very little effect overall on the community. I explain this in another slide. We did this with this guild I just mentioned or showed you before with the Mucinigenibacter Cyanophaga guild. And again, I showed you before we removed Mucinigenibacter. We see an increase in Cyanophaga and Burkholderia. We now add Mucinigenibacter and we can see Cyanophaga goes down. We add Cyanophaga, we can see Mucinigenibacter goes down. If we remove the entire guild, so this function is not present now in the synthetic community anymore, there's zero change. So no one of the other guilds care what these two organisms perform because they are not carrying out similar functions. Okay, so we could see that these probiotic interventions work and we can modify these community composition very in a targeted way by adding their competitors. The next step we wanted to do is adding metabolite. So this is more like a prebiotic intervention where we add a metabolite and increase certain members. So the idea here is we feed the competitor and by this way removing one organism out of the equation. So in order to do this, we wanted to see what these organisms are actually eating. So we looked at all the importer proteins in the DNA, RNA-seq and riboseq level. And then we looked which of these importer proteins are prioritized with the assumption that if you prioritize the import of a certain molecule, you likely want to metabolize it and eat it. So you can see here, we found 40 importer proteins which had a high translation efficiency, and the organism means they prioritize ribose import, fructose import, xylose, putrescine import. Okay, so we use this now, and this here is the organism. This is ribose importer proteins which were highly had a high translation efficiency in these organisms. And then we said, okay, if we now add ribose, we assume that any of these organisms will increase. And we did this, and what happened is that Burkholderia and Penibacillum increased in abundance. So these two organisms here, right? We could also show that this increase is concentration dependent. So when we increase the concentration, Penibacillus reacted to it in a linear fashion, whereas Burkholderia needed a high amount of ribose to be competitive. Okay. Interestingly, we also saw 3 organisms which went down in abundance. And when we look where these organisms are located, they are all located in the same kind of guild as Burkholderia. So they all are competitors and they are competing for these resources. And they are, they're basically outcompeted by the growth of one organism. We did this for a couple of other molecules. For example, what is shown here is glutathione import or putrescine import. And every time we were able to predict which organism will go up, and we could explain which organism will go down in abundance. So every arrow here is showing our prediction of organism increasing and organism decreasing. Okay, so we tested this total for 11 different metabolites, and we could, with like 80% accuracy, predict which organism will go up, and 70% accuracy, we can predict which organism goes down. And it even worked not only for organics, but also for inorganic, where we added a sulfate or thiosulfate. Okay. One comment about these experiments and also comment about actinic work with the community work. So originally, I already said at the beginning of the talk that the organism wants to do something different when in the community than when it's doing this in isolation. So when we grew these organisms on ribose, for example, 12 of these cultures, they were all able to grow on ribose actinically in a pure culture, right? That's the only food they have, so they better eat it. When we put this in a community, then we saw that 5 organisms, 4 had multiple genes turned on. 1 organism only had one little expression of a part of his transporter, but 5 organisms tried to grow on ribose in a community. Then actually at the end, only 2 organisms succeeded. So 12 organisms have the potential, but 2 are actually doing this in a community. And this is something which is extremely hard to predict if you just have pure culture data, because they will behave very differently when growing by themselves than if you grow them in the context of other organisms. Okay, so what I showed you so far is that we can modify the composition using probiotics, so adding organisms. We can modify the composition by adding prebiotics. So this was all done in a synthetic community, so this is pretty complex, but it's not as complicated as the natural soil. So we wanted to see, can we do the whole thing by studying this community in soil? So we went back to where we originally isolated some of these organisms, which is this Rhizosphere from switchgrass plants out in Oklahoma. And we got these samples through our collaborator, Tom Jünger. And then we tested a lot of different things in these communities. So we put the soil in a bucket. We added either a prebiotic, we added a probiotic, meaning a single organism, or we added the whole consortium, the 16-member consortium back, or we added combination of probiotic and a single cell, or probiotic and the whole community back. And then we studied this and see what happened. So the data looked like this, and I will simplify it in the next slide, but basically, again, this is the soil plus the syncom, and this is the soil plus syncom plus fructose. And what is highlighted here in color are the syncom members, and everything here in gray is all the other microbes in soil. So this gets very complicated to look at. So for simplicity, I remove all the other soil members right now, but they're still there in the background, right? So everyone, all the experiment is done in soil, but if I take them away, then this is how it looks. So this is the soil sample, and here we added artificially Burkholderia. So this is the control, and by adding Burkholderia, you can see we increased it just by adding it 0.3%. Okay, that's what we wanted to see. So then we added fructose because we were predicting that fructose will actually increase these organisms, and by doing so, we could increase Burkholderia now 4-fold. And surprisingly, we saw again that its competitors, Rhizobium, went down in abundance, and so did Variovorax, another competitor we predicted to go down. When we added fructose only, so now there's no Burkholderia, it's just the soil plus fructose. Again, we were able to increase Burkholderia as we predicted by over 5-fold. And again, funnily or not funnily, his competitor Rhizobium again went down in abundance. We did this for these Cytinophaga as well. We knew when we add Cytinophaga itself, it increased already 12-fold because it was a very low abundance in the natural soil. But when we then added glutathione, a molecule which we knew will increase its abundance in the syncom, again, we show an increase of almost 38% of these organisms. And its competitor, Musaligini goes down abundance in the soil. When we only add glutathione, we didn't see much change, likely because this organism is so low in abundance that the sprinkling this in would not yield too much change. Okay, so we did this for a whole bunch of different test cases. And we tested, can we predict which organism goes up, the primary target, and can we predict which organism will go down, our secondary target. So when we only add a probiotic, a single strain, we put this in, we saw that 4 out of 7 times we could predict what will happen, which organism will increase, but every time if it worked, we were 100% right that the organism we predicted also will decrease. When we did a prebiotic treatment, so now we sprinkled in some fairy dust, which we predicted, 6 out of 7 times we were able to predict the primary target, and 6 out of 6 times, 100%, we were able to say which organism will go down. When we did this now with a probiotic and a prebiotic, we were also very successful to predict these outcomes. And when we did the whole consortium and the prebiotic, every single time we were able to predict what will happen, which organism goes up and which organisms are going down. And this, in the context of a soil sample, was very exciting to us, because now we can actually go out and change certain microbes and increase microbes we deem to be beneficial in their natural environment. Okay, so I showed all this data to the syncom in soil. Now, the last couple of slides, I showed you what can happen if you go from the syncom in soil to soil sample itself. So we did all these guild analysis based on real soil samples. So we analyzed all the organisms in the soil. We were very interested in this group here containing Clostridium, Streptomyces, and Azospirillum, and we looked who are the competitors of Clostridium, and we could see that indeed the competition score showed us that Bosea, Azospirillum, and Streptomyces are strong competitors against Clostridium. So based on our predictions, we said, aha, so if we now add ribose, we knew that Clostridium will go up, will their competitors go down? And we could see that both Bosea and Azospirillum went down in abundance. And this was surprising because Azospirillum is one of the most abundant organisms in this sample. So even by increasing the Clostridium, we could push the Azospirillum numbers down. The Streptomyces was not significantly in this case. And we did the same with putrescine. Our prediction said if we add putrescine, actually we will promote the growth of Azospirillum. So in this case, the Clostridium will not increase in abundance. So we did this for a whole different organics. We added a lot of different stuff to the soil, and every time we could see if Clostridium increases in abundance, so this the fold change goes up between 5 and over 100, like 100-fold, every time Azospirillum kind of decreases in abundance. And we could see the same effect of Clostridium going up, Bosea going down, and not as strong, but still significantly for Streptomyces. When we look at all these different groups, what they are, we find that every time we add sugars, it really benefited the Clostridium. Every time we added non-sugar molecules, then the other organism benefited in this case. Okay, so last few slides. One of the things, and I spent the last 10 years of my career looking for collaborative action in different kind of microbe or microbe-host interaction. And we were very curious to see how many of these interactions we actually can explain with competition versus collaboration. So we looked at all our experiments we performed, and we found that 81%, so over 80% of our interactions, what we see can be explained by competition, whereas only less than 20% could be based on collaboration. So we don't know if this is collaboration, but this could be the case. So the possibility of collaborating collaborative organism in this soil environment is less than 20%. Okay, so I hope I showed you in this presentation that we could address now some of the major questions in microbiome research. Is basically how this community responds to perturbation and can we predict these outcomes now. And just a last slide, this method is very applicable to a wide range of samples. So we have done this already for a lot of different sample types, not only soil and the rhizosphere. And I know that you guys are not interested in human microbiomes very likely, but it also works very well. We can do this in human stool, and we have done studies already where we can modify the human microbiome targetly based on our guild and mind prediction. Okay, summary. So I showed you that we can now predict the community function. I told you about how to measure translation efficiency. Then we can use these metabolic niches in guilds. We can predict this. We can identify this interaction, in this case competition, and we can really use this to design interventions. And so this allows us now to change and engineer microbiomes. We can do this on a organismus level, basically using probiotics. We can do this on a metabolite level using prebiotics and in combinations of thereof. And this technology is really scalable, so you can do this for a wide range of microbiomes. And we already filed a patent protection for this and are in the process of spinning this out into a couple of companies. Okay, so if you missed most of the talk, two take-home messages for you. One is that microbial niche determination can predict outcomes in complex communities, and this mind and guild association identifies intervention strategies to selectively alter the microbiome. Okay, most of the work is so the work is under review right now, but we put out a preprint, which is here, and I would like to highlight all the fantastic work done in my lab, and specifically a postdoc in my lab, Oriane Moyne, who did an excellent work in order to make sense of all these complex and very new data. And then, of course, various funding agencies who support our work, and in this case, especially to the Department of Energy, BER, who funded these work in the Rhizosphere. Okay, and if you have any questions and you put them in the chat, I'm happy to entertain these questions. I know that my email was also posted in the chat earlier, and I'm happy if you don't get to your questions, shoot me an email. I'm happy to address these offline later on. Okay, thank you.
Thank you very much. That was a beautiful talk and lots to think about there. We have a number of questions rolling in, but because I am the MC, I get to start with my own question. The setup that you have in your cultures and even in the soil really forces nutrients to be the primary driver of guild formation. And I'm just wondering if you can comment in a rhizosphere situation, perhaps where you might have motility, how do you envision other factors driving guild formation?
So I think, like I said, this was the first part of the process. We are studying this already in the context of plants. So we have a bunch of plants we grow in the laboratory together with the synthetic community, and then also with soil. I think that and we study in this case the exudates of the plant, so how the plants feed certain kind of microbe they want to have, and so we in this case the majority of the interactions I think looks like it. These guilds are often driven by really organic resource or inorganic resources. The physical factors, I'm sure they play a role, but we were surprised that we could still explain a large amount of this competition having this in soil. But you're right, motility makes a big difference. So of course, if I'm a microbe which is close to the root in the rhizos plane or far out where you have no effect, I think you have to study very detailed the spatial effect, right? So you want to understand what happens around the roots and then what happened half a meter, 2 meters away. And these effects are likely very different because the plant, the exudates have, of course, an influence on the organism close to the plant and not too far away, because diffusion makes a difference.
Thank you. So I would like to encourage all participants to continue to put your questions in, and I will select questions and share them with you. One is, do you have a feel for the potential impacts of fungal and phage populations to impact the Rhizosphere bacterial communities?
Yeah, so we have, as you could see, there was not a lot of, there was not a single fungi in there. We know from previous work we have done on artificial lichens, where we work with fungus and then algae together, that of course these organisms interact in a similar way. This method could be applied to fungi as well. So we are in the process of, spinning this out to include fungi. The same is for the phage. I have, so since this is recorded, I will keep my answer very, polite. I have not seen a lot of effects on phages in our samples. This doesn't mean I know that the phages exist, and all of these organisms have prophages in their genomes, but I have not seen a lot of drastic lytic effects where the phage really ripple through the system and kill a bunch of organisms. Doesn't mean it does not exist, but we have not seen it in our systems yet.
Thank you. Another question, what will be the effect on the plant when there's an increase in the primary target by either addition of probiotics or prebiotics?
Yeah, so this is an excellent question. That's exactly what we try to address together with Trent Norton at the LBNL and John Fogel at LBNL, as well as Emily Godin at UC Davis. So we are very interested in understanding the effects between microbes and plants. Many of these molecules might, so there are two different questions. If you just add a probiotic, what people have done before, you can increase certain population. And now, for example, you know that certain microbes are always associated with higher yields of the plant. You try to promote these plants, promote these organisms. Ultimately, you want to have the plants feeding these microbes directly. So in this case, you could think of a fertilizer, like a prebiotic, which is easily distributed in the rhizosphere, will promote your plant promoting organism, but that this molecule would not interfere with the plant directly. I think it comes down to testing. You have to test if this molecule would be absorbed by the plant or not. Some of them are, some of them are not. And it depends on the time of the day, right? The plants have very different metabolism during the day and during the night. So part of our effort to look for the diurnal cycle and see how this is changing during day and nighttime. And so there are a lot of different aspects, but I'm very optimistic that this can be done in a meaningful way, and meaning that it can be translational into the field and into ultimately into farming.
Thank you. A number of people are interested in the criteria that you use to form your model syncom.
Okay. So this model syncom, we looked, and this was done by our collaborators at LBNL, Kate Salina. She has isolated close to 1,000 different microbes from the rhizosphere of switchgrass. And she looked out for organisms which are very commonly found among many, many grasses. So not only switchgrass, but they are found in the rhizosphere of all grasses. We wanted to have a community which is diverse, phylogenetically diverse. So we picked different phyla and different families. And we started off with, I think, 25 members we mixed together. There's a lot of effort going into this, how you can mix them together. We arranged for different ratios of these cells. So if you mix them 1 to 1 to 1 to 1, you get a given community. Then you mix them 1 to 10 to 25 to 28. And so the paper I listed earlier from Joanna Coker in my lab, published last year in M Systems, explains all these criteria and what we use to make this community stable and to make it reproducible, so that you always get the same kind of growth by mixing these community good together. I think that's an excellent question, and a lot of this work is based on the leg work we did a priori with all the synthetic communities. Yeah, that's a very good point.
Thank you. Another question was, were the prebiotics selected on the basis of the root exudates? And if so, then apart from sugars, should we be looking at the effects of organic acids and amino acids?
Yeah. So these probiotics, in this case, they were only selected based on what these organisms wanted to eat in this condition when we threw that in the lab in diluted R2A media. When we do this now with the root exudates, you are absolutely right. You will look for the organism will do something else when we put them together with the root exudates or with live plants, and then they will likely prioritize maybe the incorporation of certain organic acids. Root exudates contain a lot of organic acids. They also contain alcohols and a lot of variations of this. And together with Trent Norton at LBNL, we are looking at the exudation. And together with John Vogel at LBNL, we're looking at different plant lines of, in this case, Brachypodium, how we can use different mutation in transporters to study different exudation patterns, which then ripples to the effect of different microbiomes. But yes, this is correct. So this is not, what I showed you is not tailored towards root exudates. This is what we are doing, yeah, right now. Yesterday, we got a bunch of data back where we grow all the communities in ecofabs or controlled ecosystems. We run in the lab and perform analysis how they behave in and on the root.
Thank you. I'd like to remind all the participants that we won't have time to get through all the questions, and Dr. Zengler is willing to, I believe, answer some of the questions in writing afterward, and they will be posted with the YouTube. I think we have time for one more question. There's a couple people who are interested from the perspective of biological control of plant pathogens, and one of the questions is, if you notice whether the competitors are normally genetically related, and are the beneficial bacteria competitors of possible plant pathogens or of other beneficial bacteria?
I think this is exactly where we go with this, on the plant side, also on the human side, because we believe, and we have already done experiments in like mouse models with pathogens to show that we can use their natural competitors of pathogens to reduce the pathogen load. So I'm sure this also relates to plant pathogens. So the idea here is that we take advantage of competition which has been evolved over hundreds or millions of years, and that this competition, you basically feed the natural competitor. You're not going out and put any kind of drug in there where you do not know the side effects, or with standard antibiotics, you kill everyone. This is a very targeted approach where you feed natural competitors. Our hope is that the development of resistance of this will be very low, because these organisms figured out how to keep each other in check. And this is exactly the road we are already on, and studying this in the context of different diseases. Yes, this is an excellent question, and I think that we hope that this has a big. I'm very confident that this will help, and our preliminary data shows this will help us to prevent diseases. Will it cure acute diseases? So you have already an outbreak of, I don't know, let's say citrus greening or any kind of plant diseases, or this might be different to address. But I'm very positive about that this technology can be used to prevent certain kind of diseases by keeping all the pathogens in check. Then on your body, you have a bunch of pathogens, and doesn't mean you get sick or you have an infectious disease all the time. Your natural microbiome and your microflora takes care of the pathogens, and that's what we try to intend to promote with this technology.
Thank you very much. It's hard to see how this technology won't actually reach microbial outcomes that are quite diverse. I mean, there's disease, but they have influence on so many other things, as those who work in microbes know. So we really appreciate your sharing this work with us, and I hope that everybody who wants to can go back and look at the YouTube channel if you want to rewatch this, and look out for our future Phytobiome Alliance seminars and webinars. Thank you.
Thank you so much. And if I didn't address any questions, I'm happy to answer them. Might not happen in the next 5 minutes, but I try to get to this as soon as I can. Okay, thank you.
All right.